Hello friends, welcome to Biology Made Easy. Today I'll be bringing you a very intriguing topic known as the hypoxia inducible factor, which was also responsible for the bagging of the Nobel Prize in 2019 for Physiology and Medicine, which was bestowed upon three great scientists, William G. Crellin, Peter J. Ratcliffe and Greg L. Semenza. Their primary work was on the unveiling of the pathway of hypoxia inducible factor. Hypoxia inducible factor, as we all know, has pretty huge implications on various pathways like inflammatory responses, fatty acid metabolism, glucose metabolism, cancer metabolism, right? Even in mitochondrial dysfunction, etc. Now, what is HIF, hypoxia inducible factor? It gets induced during hypoxic or when the cell is under lesser oxygen concentration than it is required for the cell. Now, HIF is a helix loop helix transcriptional activator. Activators are really responsible for the recruitment of RNA polymerases via after association with coactivators and mediators. Now, it's a helix loop helix transcription factor and it's a heterodimeric transcription factor, meaning that the two subunits would not be same, they will be different. One of the subunits could be HIF1 alpha, HIF2 alpha or HIF3 alpha. The other subunit could be your HIF1 beta, also known by the name ARNT, aryl hydrocarbon receptor nuclear translocator protein. Now, HIF1 alpha is a constitutively active and expressed transcriptional factor with wide array of expression. Now, HIF2 alpha is only expressed in myeloid derived cells and vascular endothelial cells, whereas HIF3 alpha is a pretty weak transcriptional activator. In this video, we will be focusing solely upon the HIF1 alpha pathway. HIF1 alpha is made up of two TAD domains. TAD means transcriptional or transactivation domains or transcriptional activation domains. The TAD domains could be subdivided into two parts. One part is NTAD, other is the CTAD. NTAD is known as the NH2 or N-terminal transactivation domain, whereas the CTAD is known as carboxyl COOH or the CTAD, C transactivation domain. The NTAD coincides with yet another specific domain known as the ODD domain, which is of huge implication, known as the oxygen-dependent destructive domain. Now we will be shifting our focus to the signaling of the hypoxia inducible factor. There are many pathways, PVHL dependent, independent. In this video, I will be talking about the protein wall Hip-Lindea or von Hippel-Lindea protein dependent pathway, which controls the upregulation and downregulation of the HIF1 alpha. Now what happens is, if the cell is hyperoxic, means it has got more oxygen than it requires. At that point of time, there are proteins known as PHD, prolyl hydroxylases, right? So these proteins are activated due to the extreme oxygen concentration, and what they do is they then are able to hydroxylate the proline at 402 and 564 residues present in the oxygen dependent destructive domain in the NTAD of HIF1 alpha. After the hydroxylation takes place, we go to acetylation phase. Acetylation is not directly dependent upon the oxygen concentration, but once the hydroxylation takes place, acetylation usually follows by a temporarily unknown mechanism. Now, acetylation is preferred or is catalyzed by the ARD1 or arrest defective 1 protein, and once this acetylation and hydroxylation or addition of OH groups are done, what happens is specifically the hydroxylation is responsible for it to be recognized by a specific protein known as the von Hippel-Lindea protein. Once von Hippel-Lindea protein recognizes the hydroxylated proline residues present on the ODD domain of HIF1 alpha's NTAD, what happens is it becomes primed, it becomes tagged to be recognized by the E3 ubiquitin ligases, mostly elongins. Once E3 ubiquitin ligases recognize it, we all know that it will go for then ubiquitination. It will be recruited, it will be shifted to the 26S proteasome in the eukaryotes, and then the protein would be cleaved, also known as ubiquitination pathway. Now, one more thing is there is a residue known as asparagine at 803. That is also hydroxylated by asparagine hydroxylases, and when that happens, the HIF1 loses the ability to bind with the coactivators. This process also takes place once the oxygen concentration is too notched up or too enhanced in the cell. Now, if the oxygen concentration is less than required, means the cell is under hypoxic condition by the name hypoxia inducible factor. In the hypoxic conditions, the HIF1 alpha expression would be induced. How? Because since the oxygen concentration is low, the PHDs or prolyl hydroxylases would not be activated. I have already explained to you that activation of the PHD is directly proportional to the concentration of oxygen. If the concentration of oxygen is pretty low, it won't be activated. Since PhDs won't be activated, it won't be able to hydroxylate the proline at 4O2 and 564 residues. Since the hydroxylation process cannot take place, what would happen is your von Hippel-Lindau protein would be rendered unable to recognize the O-triple-D domain of the NTAD of HIF1 alpha. Since this von Hippel-Lindau protein cannot recognize the O-triple-D domain, it cannot really bind. Therefore, what would happen is the E3 ubiquitin ligases like the elongins would be again unable to recognize the O-triple-D domain. And since they cannot recognize this very domain, what would happen is the 26S proteasome cannot degrade the HIF1 alpha, and HIF1 alpha expression would be induced incessantly till the oxygen level again gets notched up or again increases in the cell. So that's about it in the video. I hope that you were able to comprehend the lecture. And if you have any queries, any doubts, kindly don't hesitate to put your questions in the comment section below. If you really find the content relevant, then do like the video, and please also hit the subscribe button and the notification bell so as to be notified whenever my next video comes online. Thank you.